mastigophora, also known as metamonada, are both words that are used in scientific classification. They represent phyla of flagellated unicellular eukaryotes. We often find that the example organisms that we're going to use that are flagellated unicellular microorganisms, Giardia lamblia and Trichomonas vaginalis, might be classified in either phylum. Metamonada is the more modern classification scheme, but there are still quite a few parasitologists that still recognize mastigophora as a phylum as well. As you may recall, these unicellular eukaryotes are quite diverse, and there's a bit of a challenge to The challenge is, of course, trying to determine through scientific means which group or which phylum to classify some of these organisms. Oftentimes, the older classification schemes, like the mastigophora, simply relied on motility structures, like flagella, and didn't look at more detailed descriptive characteristics. That's often why some scientists still use the older classification schemes. They're simply simpler. Giardia lamblia can be found in two forms or structures. One is a trophozoite, which simply means it only has a cytoplasmic membrane as its outer surface, and it has a teardrop shape with two nuclei, so that when you look at these through the microscope, they often represent or appear to be looking back at you with those two nucleated cells. That very distinctive teardrop shape, it's hard to miss when you're looking through a stool sample that's been properly prepared, and you can find those tiny little single-celled organisms in the different fields of images when you're looking through the microscope. Now, the infective stage of Giardia lamblia is actually a cyst stage. The cyst of Giardia has a hard outer covering. That hard outer covering allows this parasite to tolerate changes in the environment, drying, changes in nutrition, maybe nutrients have sort of gotten less available. So anytime that there's an environmental challenge, it's not unusual for Giardia to change from the trophozoite form to the cyst form. And it's the cyst that we ingest, gets into our intestinal tract, and causes severe gastroenteritis, or enteritis alone, which would be intestinal upset. Lots of cramping, pain, and diarrhea. There are various names for the intestinal upset that Giardia lamblia can cause. It is one of the easiest to identify in a stool specimen because of its distinctive shape. It is an archaeozoa. It grows under anaerobic conditions and does not have any intracellular mitochondria or intracellular bacteria functioning like mitochondria. And names for the condition that it can cause: backpacker's diarrhea and beaver fever. Trichomonas vaginalis is another example of a mastigophora, or organism classified in mastigophora or metamonada, and is the common cause of protozoan vaginitis. So vaginitis caused by a protozoan, it's Trichomonas vaginalis that's the most likely cause. Now, there are other causes of vaginitis. It can be due to yeast, bacteria, and perhaps even viruses. But if we're talking about protozoa, we're talking about single-celled eukaryotes, it's Trichomonas vaginalis that we think of first. The clinical name, if we don't want to just have the general terminology of vaginitis, which is inflammation of the vagina, we could more specifically say that the patient has trichomoniasis. There is no cyst form, and in fact the infective stage of Trichomonas vaginalis is the soft-sided trophozoite form. A trophozoite is simply a single-celled organism that has no shell or hard outer covering and just has the cytoplasmic membrane as its outer surface. You can see the pairs of flagella sticking out of one spot on the surface of the single-celled organism, and it does swim around quite quickly in a moist liquid environment or a moist, essentially, surface of a mucous membrane, and it is classified as a sexually transmitted disease because it can be transferred from one person to another when we have close physical contact. For the identification of trichomonas, if you look on many requisition forms in doctor's offices or clinics, you'll find that one test that can be checked off, essentially looking for these types of protozoa, is called a hanging drop. A hanging drop, quite technically, is where we would take a specimen by collecting with a sterile swab from the patient's cervix and vagina. We would mix the scraping of those cells from those tissues or surfaces with some isotonic saline. Then we'd take a drop of the mixture of the vaginal scrapings in isotonic saline, place the drop on a cover slip, and then literally suspend that cover slip in midair on a microscope slide so that we would allow any kind of protozoa or organism to freely swim around and more easily be identified in the mixture of bacteria and normal healthy cells from the vaginal or cervical area to float around in that isotonic saline. Any kind of parasite that has flagella will be able to swim quickly and easily in this hanging drop. Now, most of the time when we order a hanging drop, that's really not what gets performed in the laboratory. Technically, a hanging drop is very challenging, believe it or not, and it's simpler and quicker and easier to simply make a wet mount of the same mixture. Take a drop of the material scraped from the affected area on a clean microscope slide, add atop that drop a nice, clean, clear slide cover or cover slip, and then take a look, and again, any kind of flagellated, ciliated protozoan that's moving around by other means, other than just sort of drifting around in the liquid, can easily be seen even in a wet mount. Apicomplexa is the next phylum where we find unicellular eukaryotes that have no motility structures. Two distinctively different genera, Plasmodium and Toxoplasma, are good examples of eukaryotic organisms. We have the genus Plasmodium and 4 species of Plasmodium. Plasmodium vivax, which is the most common form of tertian malaria, T as in Tom, ER, T as in Tom, IAN. The symptoms of tertian malaria occur every 48 hours. The second species of Plasmodium is Plasmodium falciparum. Plasmodium falciparum is the most lethal form of malaria. We often call it blackwater fever or tertian malaria with symptoms every 48 hours, but much more severe than vivax malaria. Plasmodium malariae, which is quartan malaria with symptoms every 72 hours, which is much more rare than the first 2 types of malaria, tertian due to vivax and tertian due to falciparum, and Plasmodium ovale, which is the 3rd type of tertian malaria, again much more rare than Plasmodium vivax or falciparum, with symptoms every 48 hours. The second genus in Apicomplexa that we need to talk about is Toxoplasma, and we'll see more information about Toxoplasma in the next couple slides. Plasmodium species cause malaria. The symptoms of malaria are fever and chills. The chills are so severe, the patients have been known to shake themselves off the bed. And the fever, there's very profuse sweating that occurs. During the febrile stage, very profuse sweating occurs to the point with a tremendous loss of body fluid. And in fact, the Plasmodium parasite grows and lives inside the red blood cells and the cells of the liver of the patient that's infected, consuming the intracellular contents of those cells in order to reproduce and continue to grow. Here's our first example of a biological vector, which would be a biting mosquito or a biting insect. Biological vectors in general need a blood meal as part of their egg manufacturing, egg-laying process, and include not only mosquitoes, but other biting insects like ticks, fleas, lice. But for the transmission of Plasmodium parasites, we need a mosquito. And typically a mosquito that likes very quiet, still night air. Often that kind of still air that we find inside of, let's say, a bedroom or a porch or some kind of housing area, not out in the woods. And so typically we find the mosquitoes that are often going to transmit Plasmodium are ones that often live close to or with human populations.